Hello everyone, I am Yushita. Welcome to my YouTube channel. Today I am going to discuss about polymerase chain reaction. The contents that I am discussing are principle of PCR, PCR steps, types of PCR, applications of PCR. Now discuss firstly about its introduction. PCR or polymerase chain reaction is a technique used in molecular biology to create several copies of a certain DNA segment. This technique was developed in 1983 by Carey Mullis, an American biochemist. PCR has made it possible to generate millions of copies of a small segment of DNA. This tool is commonly used in the molecular biology and biotechnology labs. Now coming to the principle of PCR. The PCR technique is based on the enzymatic replication of DNA. In PCR, a short segment of DNA is amplified using primer-mediated enzymes. DNA polymerase synthesizes new strands of DNA complementary to the template DNA. The DNA polymerase can add a nucleotide to the pre-existing 3' OH group only. Therefore, a primer is required. Thus, more nucleotides are added to the 3' end of the DNA polymerase. And now coming to the methodology of PCR. The PCR involves three major cyclic reactions: denaturation, annealing, elongation. Denaturation occurs when the reaction mixture is heated to 94 degrees centigrade for about 0.5 to 2 minutes. This breaks the hydrogen bonds between the two strands of the DNA and converts it into a single-stranded DNA. The single strands now act as a template for the production of new strands of DNA. The temperature should be provided for a long time to ensure the separation of the two strands. This is the basic methodology of the polymerase chain reaction. First is the denaturation, and then annealing, and then elongation. This is the diagrammatic representation of the process. And then the second step of the PCR is annealing. The reaction temperature is lowered at 54 to 60 degrees centigrade for around 20 to 40 seconds. Here the primer is bind to their complementary sequences on the template DNA. Primers are single-stranded sequence of DNA or RNA around 20 to 30 bases in length. They serve as the starting point of the synthesis of DNA. The two separated strands run in the opposite direction, and consequently there are two primers, a forward primer and a reverse primer. Now coming to the third step of the PCR, it's elongation. At this step, the temperature is raised to 72 to 80 degrees centigrade. The bases are added to the 3' end of the primer by Taq polymerase enzyme. This elongates the DNA in the 5' to T' direction. The DNA polymerase adds about 1000 base pairs per minute under optimum conditions. Taq polymerase can tolerate very high temperatures. It attaches to the primer and adds DNA bases to the single strand. As a result, a double-stranded DNA molecule is obtained. These three steps are repeated 20 to 40 times in order to obtain a number of sequences of DNA of interest in a very short time period. And now coming to the types of PCR. PCR is of following types, and the first type among them are real time PCR. In this type, the DNA amplification is detected in real time with the help of fluorescent reporter. The single strength of the fluorescent reporter is directly proportional to the number of amplified DNA molecules. And coming to the next type of the PCR is nested PCR. This was designed to improve sensitivity and specificity. They reduce the non-specific binding of products due to amplification of unexpected primer binding sites. The third type of the PCR is multiplex PCR. This is used for the amplification of multiple targets in a single PCR experiment. It amplifies many different DNA sequences simultaneously. And coming to the next type of PCR is quantitative PCR. It uses the DNA amplification linearity to detect, characterize, and quantify a known sequence in a sample. And coming to the next type of PCR, it is arbitrary primed PCR. It is a DNA fingerprinting technique based on PCR. It uses primers, the DNA sequences of which is chosen arbitrarily. Now coming to the next type of PCR, that is hot start PCR. It is a modified form of conventional polymerase chain reaction that reduces the presence of undesired products and primer dimers due to non-specific DNA amplification at room temperatures. And then touchdown PCR. It is a method to reduce off-target priming and hence to increase the specificity of PCRs. In touchdown PCR, the temperature selected for the annealing step is initially set 5 to 10 degrees centigrade higher than the calculated TM of the primers. And now coming to the applications of the PCR. The following are the applications of PCR. In the stream of the medicine, testing of genetic disease mutations, and it also used in monitoring the gene in gene therapy and also used in detecting disease causing gene in the parents. And coming to the stream of forensic sciences, used as a tool in genetic fingerprinting and also used in identifying the criminal from millions of people and also used in paternity tests. Coming to the stream of research and genetics, it used it to compare the genome of two organisms in genomic studies. It's also used in the phylogenetic analysis of DNA from any source such as fossils. Also used it to analyze of gene expression and also used in gene mapping. Thank you. Thank you for watching this video.